Okay, we're back. And it should be 203 nerve 3. And this, we should be able to finish chapter 12 with this video. I think we left off at events in each section, the receptive segment. Remember what the receptive segment is? It's the dendrites in the soma, or cell body. And they receive information, either sensory information from outside or in your body, or from another nerve cell. And what happens in there are postsynaptic potentials. These are graded potentials. And so we're talking about the postsynaptic cell. There are two types of graded potentials, EPSPs and IPSPs, excitatory postsynaptic potentials, inhibitory postsynaptic potentials. And so an excitatory postsynaptic potential is going to depolarize the postsynaptic cell. The inhibitory postsynaptic potential is going to inhibit. It's going to be a hyperpolarization. So EPSPs bring a cell closer to firing, turning on. IPSPs bring the cell farther away from turning on. So what's important is whether a cell is turned on or not. When I say turned on, I mean there's a fire and action potential. That's on. And whether it does or not is dependent upon the membrane potential at the initial segment, or the axon hillock. And these postsynaptic potentials are going to summate, add together, and if they bring the initial segment to minus 55 millivolts, from minus 70 to minus 55, that's called threshold. And then an action potential will fire. And we're going to look at the events in an action potential in a little bit here, and what ion channels are turned on, which ones are turned off, which ones are inactivated. All these EPSPs and IPSPs are all added because they travel from where they happen on the dendrites to the initial segment, right? And they get smaller the farther they have to go to get smaller. And remember the characteristics of graded potentials or local potentials. And so if these IPSPs and EPSPs don't bring the, particularly APSPs, don't bring the polarize the membrane potential at the initial segment to minus 55, that's called subthreshold. And that means the cell's off. If it does bring it to minus 55, then it is threshold, and the cell will fire an action potential. Okay. And there's two types of summation, spatial and temporal. Spatial is two synapses going off at the same time in different parts of the cell. And when they move down the dendrites and hit this soma, they add together to be a larger one. Temporal is if you fire one EPSP, and then before it, so you get this blip, depolarization, and before that goes back to membrane potential, another synapse fires, then that's called temporal, and that will add upon each other. So they'll make bigger depolarizations. And also, IPSPs will cancel out EPSPs. If you get an EPSP firing off at a particular size, and at the same time, you have an IPSP going off at the same time. When they get to the soma, they'll cancel each other out, and nothing will happen. So they'll cancel each other out. So the membrane potential of the initial segment is bopping up and down. This depending upon how many EPSPs and how many IPSPs are going off, and where they are in a cell, and how close together they are, and whether they're doing in rapid succession. And And like I said, if you depolarize the initial segment to -55, you'll fire an exponential. So closer to the initial segment, more effective. Because remember, these are gradient potentials. They decrease in size. So ones that are closer to the initial segment are going to have a greater effect on whether the cell turns on or not. Okay. So I just mentioned this way, sub-threshold and subthreshold. An AP is generated threshold. And it's just the all or none law, because remember, action potentials are really different from gradient potentials. They are either turn on completely or don't turn on at all. And they're always the same size. They're stereotypical. Every action potential looks exactly like every other action potential. And that's called all or none law. Either you get a full action potential or you don't get anything. You don't get halfway or 3/4. Okay, in the conductive segment. So this is the where the action potential is conducted and the axon, propagation of the depolarization. And what this nerve signal is, what the action potential is just a brief reversal of charge of the cell membrane. It becomes positive inside, and then becomes negative again. And this positive flipping moves down the axon and goes on to the axon terminals. Okay. So propagation of depolarization, propagation of repolarization. So you're depolarized first, and then you're repolarized. So you start off negative, you go positive, you go back to negative. And this brief chain reversal of charge moves down the axon. We'll show you later on how it moves down the axon. Okay, and it's after hyperpolarization. After the repolarization, the cell becomes slightly hyperpolarized. It's called the active hyperpolarization. And then it quickly returns to the resting membrane potential. And there are two refractory periods. And this is why action potentials only go in one direction, because the immediate part of the axon that's behind, so an action potential is traveling along, and the part that's behind, here's the action potential, and as it moves down, the piece of axon just behind it is in what's called absolute refractory period, where sodium gates are inactivated and you cannot generate another action potential. So this is why action potentials only go in one direction. Gradient potentials go in both directions. Action potentials only go in one direction. And here, this is right out of the book, tells you what's happening here. So you're at a resting membrane potential here, and there are EPSPs bopping along, and they're summating together, and there's enough of them where they get the membrane potential at the initial zone to -55. And here we get the depolarization. When threshold is reached, voltage-gated sodium channels open up, and explosively, they go from closed to wide open for a brief fraction of a millisecond, and then they close. And so what's happening is sodium is rushing into the cell, and sodium is trying to get to its equilibrium potential, which is +60. Doesn't quite get there though. It's only open for a brief fraction of a millisecond and then closes. Now when it closes, it becomes inactivated at about +30. So the polarization phase gets to about +30. Then sodium gates are inactivated. And sodium can't rush in anymore. At that point, potassium gates have been opened. Now potassium gates open up, they're delayed a little bit. First sodium gates open up, and then actually near the top part of the depolarization phase, potassium gates open up, and potassium starts to rush out of the cell. But it does so slowly, and it doesn't have too much effect. until the sodium gates are inactivated at about plus 30, and now these potassium gates are wide open, and potassium just rushes out of the cell, brings positive charge with it. So that's the repolarization phase 4. Voltage gated, the voltage gated sodium channels are inactivated, opening, and the voltage gated K channels are wide open. K moves out of the cell and brings the membrane potential back into negative territory. And the thing is, potassium gates are slow to open, and they're also slow to close. So they get back down to minus 70, but they're still partially open because they haven't quite closed yet. So a little more potassium leaves than should, and you get this what's called after hyperpolarization. But then the potassium gets closed, and we get right back to resting membrane potential. And what happens here is that when the membrane potential gets back to minus 70, sodium gates change from inactivated to just closed. And remember what the difference there is. The difference between inactivated and closed is that closed, you could reopen them. Inactivated, you can't. So this whole repolarization phase from here down to here is called the absolute refractory period, because sodium gates are inactivated and nothing is going to open them up. Now the after hyperpolarization, right down here, is called the relative refractory period, because sodium gates are now closed, which means they can be reopened, but they're farther away from threshold than normal. So you're going to need more depolarization, more EPSPs going off to fire another action potential at this time, because you're farther away from threshold. So you need more activity in EPSPs. Okay. So synapses. We're going to talk more about synapses, the structure and function of synapses, of chemical synapses. And neurotransmitters. Remember how the arrival of an action potential at an axon terminal will cause a release of neurotransmitters in the presynaptic membrane. They'll diffuse across the synaptic cleft, bind to receptors on a postsynaptic membrane, and these receptors are associated with ion channels, and they're open ion channels. And they'll have an effect on the membrane potential of the postsynaptic cell. And here's the thing. If neurotransmitters open up sodium ion channels in the postsynaptic cell, you're going to get an EPSP. Because sodium will rush in. If, however, the neurotransmitter receptor is associated with a potassium channel or a chloride channel, you're going to get an IPSP, an inhibitory postsynaptic potential. Because if you open up potassium ion channels, remember what happens. Potassium rushes out of the cell, brings positive charge out, and it becomes more negative inside. Chloride works in the opposite way. Chloride is more concentrated, remember, outside of cells than inside. So if you open up a chloride ion channel, chloride ion rushes into the cell, and it brings negative charge in, and that causes a hyperpolarization, or an IPSP. Okay, the conduction of action potentials. There's two ways action potentials get conducted. One is called continuous conduction in what's called unmyelinated axons. And that means every piece of axon has to be stimulated to threshold. Every single piece. And that's why it's a lot slower. But myelinated axons, and this is the real value of myelin action, really speeds up conduction. It's called saltatory conduction. Now, this is really hard to see here. It's small, but it's right out of the book. The nodes of Ranby are also neurofibril nodes. They're called neurofibril nodes. I learned them as nodes or NVA, but they're getting away from eponymous names for stuff now. Anyway, so what happens in a myelinated axon is the exponential travels from node to node to node to node. Not every little piece of membrane is fired to exponential, just at the nodes. So sodium rushes in at the node, and sodium diffuses along the axon in both directions. And it goes over, it travels along the axon underneath the myelin sheath to the next node. And that sodium ion is going to cause cause the membrane in the next node to reach threshold, and another exponential will fire to that node. And those sodium ions will drift to diffuse up to the next node and cause that one to be a threshold, and so on and so on. But remember, the sodium ions are actually diffusing in both directions, in the forward direction toward the axon terminus, but they're also diffusing back toward the cell body. So remember, why doesn't the previous node then fire an exponential? Because the sodium ions are going there. That previous node, remember, is in absolute refractory period. So it doesn't matter how much sodium ion goes there, how much you try to depolarize it, it's not going to happen because the sodium gates are inactivated. Okay, and remember, that's why exponentials only go in one direction. Okay, so we'll go look at the sequence of events in synaptic transmission. What happens when the exponential reaches the synaptic bulb or axon terminal? We give examples of particular types of synapses, one called excitatory cholinergic. Cholinergic means acetylcholine is the neurotransmitter. Then we're going to look at inhibitory GABAergic. GABA is gamma aminobutyric acid. It's an amino acid. And here's one of the unusual amino acids. We usually think of amino acids as making up proteins. There's 20 amino acids that make up all proteins. This is not one of them. This is an amino acid that you don't find it in any protein anywhere. It is a neurotransmitter. And it's usually inhibitory, but remember, acetylcholine is usually excitatory, but you gotta remember what makes a synapse excitatory or inhibitory, not the neurotransmitter that's released. Although most GABAergic are inhibitory, most acetylcholine are excitatory, but that's not what makes them excitatory and inhibitory. It's what ion channel is associated with the receptor, like I just said a little while ago. If the neurotransmitter receptor is associated with a sodium ion channel, you're going to get an EPSP. If it's associated with a potassium or colloid channel, you're going to get an IPSP. And it doesn't matter what the neurotransmitter is. And then we're going to look at excitatory adrenergic. Adrenergic uses norepinephrine, which is a biogenic amine. And also, it's a little complicated, a little slower, because it has to use a second messenger system. Remember the second messenger system? This was very similar to how water-soluble hormones work, right? They have to bind to a membrane receptor, and they have to activate a second messenger, like cyclic AMP, that will open up ion channels or cause genes to be transcriber, so on and so forth. So, norepinephrine, it's a neurotransmitter, but it's very similar to epinephrine, which is actually a hormone, remember, from the adrenal medulla. Remember, the adrenal medulla is really nerve tissue. It's nerves. It's axonic neurons. And we talked about that in the early part of this chapter. However, the parts of a neuron, and all neurons have axons, except the ones in the adrenal medulla. They don't have an axon because they don't fire Ash potentials. They secrete norepinephrine. So they're nerve cells in your adrenal gland. So really the adrenal medulla is like a ganglion. Okay. Then cessation of signal. Just as important as turning on a synapse is turning it off. You want to turn it off. How do you turn off synapses? Well, sometimes the neurotransmitter just diffuses out into the synaptic cleft. You know, and it may be picked up by other cells, but it just diffuses out of the cleft. Sometimes it's reuptake. That is, the neurotransmitter is taken up, back up by the presynaptic cell and reused. So serotonin is one like that. That's another biogenic amine. And then sometimes it's degraded, it's broken down. And that's what happens to acetylcholine, to these cholinergic synapses, because there's an enzyme in the synaptic cleft called acetylcholinesterase, which chews up acetylcholine. So as soon as acetylcholine binds, opens the ion channels, causes a change in the membrane of the postsynaptic cell, this enzyme comes along and chews it up and stops the synapse, turns the synapse off. And one of the very first nerve gases that was developed was an acetylcholinesterase inhibitor, something that will inhibit acetylcholinesterase from being chewed up, which means the synapses don't turn off. And all of your neuromuscular junctions are acetylcholine. So you go into violent convulsions, and it's really a seriously bad death if you can't turn your synapses off. And that was one of the very first nerve gases developed. Okay, so here, let's look at what happens. One, action potential reaches synaptic knob, all right? And the synaptic knob, or the axon terminal, is depolarized. What does this do? Opens up voltage-gated calcium channels. Right? So that's important to realize. Where do you find voltage-gated calcium channels? In axon terminals. And calcium rushes, remember, calcium always rushes into cells because it's always concentrated outside than in. And what it does, well, it depolarizes the cell, but the important thing is it binds to the synaptic vesicles and causes them to merge with the presynaptic membrane. And when they merge with the presynaptic membrane, they push the neurotransmitter into the synaptic cleft by exocytosis. The neurotransmitter diffuses across the synaptic cleft, binds to its receptor on the postsynaptic membrane. That binding opens up an ion channel. Ions either rush in or out, depending upon what the ion channel is. And the memory potential of the postsynaptic cell is changed. It's either depolarized or hyperpolarized. And then the synapse is turned off somehow by those three methods we talked about, and then the synapse is off. And this happens to be a neuromuscular junction. So this is a cholinergic synapse here. So here you can have a cholinergic synapse that has different receptors. Well, we're going to get to this later on in a later chapter. I think in the autonomic nervous system chapter, we're going to talk about different kinds of neurotransmitter receptors. In acetylcholine, there's two types: nicotinic and muscarinic. Nicotinic is direct effect. Acetylcholine binds to a nicotinic receptor. It's called nicotine because it's nicotinic because it's stimulated by nicotine and also curare. And sodium ion channels are open, and sodium rushes in and causes an EPSP. Some cholinergic synapses have muscarinic receptors. These things, they're named after some kind of compound from mushrooms called muscarine. but they're also blocked by atropine. And what they do is they activate a G protein, a second messenger, that will go over and open up ion channels. So it's a little slower process, usually longer lasting though. So that's called an indirect process. But they're both excitatory because they open up sodium ion channels. Now, norepinephrine generally works with second messenger also. And GABAergic does the same thing. Well, it's more direct, I think. It binds to neurotransmitter receptor, opens up ion channels. Okay, so let's look at neurotransmitters and need to know their chemical structure and their function. So you got biogenic amines like serotonin and dopamine. You got amino acids like GABA. You got neuropeptides, which are actually small proteins, actually. You also have a gas, nitric oxide. They don't put up that this is really strange. It's a gas, very simple compound, but it acts as a neurotransmitter. And then you have acetylcholine, which is in a class all by itself. That's the chemical nature. You need to know, if I mention neurotransmitter to you, what its chemical structure is. The biogenic amine, is it amino acid, neuropeptide, or is it acetylcholine? Or is it a gas, like nitric oxide? Okay, function, effects, actions. Excitatory causes EPSB, inhibitory causes IPSB, depending upon the ion channel that's opened up. Action is directly opens ion channels, like what happens in nicotinic receptors, or indirectly cause a response to the activation of G proteins that leads to the manufacture of a second messenger, and that opens up ion channels. And that's more adrenergic or cholinergic with muscarinic receptors. Okay, then you have what are called neuromodulators. And these are usually purifiers. They're like local hormones. And some of them are released by postsynaptic cell and get diffused back and affect the presynaptic cell, go in the opposite direction. It's called neuromodulation. And this is going to cause a strengthening of synapses. And when we talk about strength of synapse, what do we mean? Well, how much effect does this synapse have on the membrane potential of the postsynaptic cell? And that's largely due to how much neurotransmitter is released. And that's due a lot to how much calcium enters the synaptic knob. And some of these neuromodulators, what they do is they cause more calcium channels to be opened up when the action potential comes down. So more calcium gets in the synaptic knob, more neurotransmitters released, and this is called a facilitated synapse. It's more effective. It has a greater effect on a postsynaptic cell. Some of these are neuropeptides, like endorphins and enkephalins. And these are the natural opioids that we make in our brain. And we always wondered, why would this plant product, from the opium poppy, have this huge effect on human brains? Why is that? It must be binding to some kind of receptor in the brain. Same thing with marijuana. We know the cannabinoid receptors. Why does marijuana have this effect in your brain? Because they're receptors, natural opioid receptors, and you make natural opioids called endorphins and enkephalins. They make you feel real good. Okay. neurointegration, postsynaptic potentials. So remember, integration is an adding. This means adding. And remember, these postsynaptic potentials are integrated at the initial segment, the axon hillock. Remember what we said, temporal and spatial summation. If they're close to each other and they fire at the same time, when they get to the initial segment, they'll add. Or if you have one synapse that fires very rapidly, it'll have a bigger effect. And presynaptic inhibition is a synapse on top of another synapse. You have one synapse, and on the side of that synapse, you have an inhibitory synapse. What you can do is turn off a synapse. If this cell fires an exponential down its axon, it causes an IPSB in a synaptic knob of another neuron. And that means when the exponential reaches the synaptic knob, the cell isn't depolarized, or depolarized much less. So there's like no neuron, so calcium ion channels are not open, calcium isn't open, and the neurotransmitter is not released, and these synapses turned off, or it's inhibited. Only a little bit of neurotransmitter is released. That's called presynaptic inhibition. And that's one way, when we get to the senses, we'll look at a pain pathway, or now I should be an analgesic pathway, a natural analgesic pathway. When you first injure yourself, the pain is intense, but slowly it degrades into like a duller, much less intense pain. And that's due to presynaptic inhibition of certain synapses in your spinal cord that release this substance, this neurotransmitter called substance P. I think it's a neuropeptide, and it's the pain neurotransmitter. And so it stops the pain synapses by presynaptic inhibition. Okay, here is a typical cell that has look at all these, here's a cell body of dendrites, and here's all these axon terminals throughout the binding all over the place, causing EPSBs and IPSBs all over the bodies itself. And here's an actual picture of a cell with all the presynaptic axons and synaptic knobs. And here's the thing. On the average, every one of your brain cells is in contact with 5,000 other cells. 5,000. And there are about 100 billion cells in your brain. Each one has on average 5,000 connections with other cells. So there are about 500 trillion synapses in your brain. 500 trillion. Wow. And so, and the more connected your brain is, the more powerful it is. Okay. Neural coding. Okay. And this is something called modality. What kind of inputs coming into your body? And we're going to talk about more about this when we get to the senses. You have all this information coming in. You have your senses. You have touch, pain, sight, hearing, smell. How does your brain can tell the difference between smelling and hearing, for example, or smelling and seeing? That sounds like a stupid question. It's obvious. Well, not when you think about it, because what does your brain actually get from the periphery? Does it get sound waves? Does it get smells? Does it get light? No. What does your brain get? Action potentials. That's all it gets. Just action potentials. And every action potential looks like every other action potential. They're identical, they're stereotypical. So vision comes into your brain as action potentials. Smell has action potential. Hearing has action potential. Touch has action potential. So how does your brain know which is which? Called labeled lines. This information goes to a different area of your brain. Hearing goes into temporal lobe. Vision goes into the occipital lobe. Smell goes into what's called the insula. Goes to a different area in the brain. And that's how your brain knows the difference between these different senses. Okay, intensity. How does the brain know a weak stimulation from a strong stimulation? A bright light versus a dull light, a hard touch versus a soft touch, loud noises, whispers. How does it know? So that's the intensity of the input, and that's action potential frequency. The more intense the stimulation, the higher the frequency of the action potentials. And this is and we used to talk about frequency of action potentials as how many impulses per second. 10 impulses per second, 100, 1,000 impulses per second. Types of neuronal circuits. Okay, I'm going to be talking about these in the next lecture. I think we're going to end this one pretty soon and then show you pictures of these types of neural circuits called diverging, converging, reverberating, and parallel axis discharge. What they look like and what people think of what their functions are, what their roles are. And then we're going to talk more about neuromodulation. So I'm going to hold that off until the next video. So I'm going to stop this one. So this is 203 nerve 3, and then we'll go on to we'll finish up the chapter with the next video. All right.